Hello. Today I'm going to do some prep for lecture 13, which is not until next week, but it's good to get it done in advance. And so the material we've been focusing on the last few lectures has been single gene crosses and using in equal segregation as the principle to generate ratios and to predict outcomes and so on. And we're starting now to look at the second law, which is independent assortment. And so this is the point at which Mendel started looking at combinations, pairs of genes together. And he determined, fortunately, that the alleles of one gene assorted into gametes, so was divided into gametes equally likely to get the dominant yellow or the recessive green allele if you had a heterozygous individual, but that the assortment of the yellow gene and the round versus wrinkled P gene did not assort with any reference to each other. So for example, the two dominant alleles didn't always go together. The two recessives didn't, the dominant of one and the recessive of the other. There was no preference for any particular combination. And so that is referred to as independent assortment. Because it's like flipping two coins. If one coin comes up heads, the other one doesn't check to see what the first coin did. It's tossed independently, and the outcome of that coin doesn't depend on what happened with the first coin. And I think for most people that's a fairly intuitive topic, but when we start talking about genes, it feels more complicated. But really, that's how it works, and it's the same kind of thinking. So Mendel happened to choose 7 different traits to study. He did study a couple of others a little bit, and it did turn out that there were some strange connections between them, and he did not use those data because he couldn't interpret them. This is apocryphal. This is probably a matter of record, but I cannot remember exactly where I read it, so I cannot say for sure it's a reputable source, because you always have to check the validity of your sources. But the ones he reported at the least were 7 specific traits, and they happen to be on different chromosomes, and the P has 7 chromosomes. And so that was a lucky thing, because his second law couldn't have been determined without that being the case. So he had studied each of those 7 traits individually, and I'll probably remind you at some point what he found about, dominant alleles, and then F1 crossed to each other, giving those 3-to-1 ratios. But then he started dealing with them in pairs, and so we're just going to look at one example. But he did this with all the different combinations. Let's do red. And so for the P generation, he crossed pure breeding, round and yellow. So the seeds can be round, or they can be wrinkled, sort of shriveled. And they can be yellow in color, or they can be green in color. And so he crossed round and yellow, pure breeding P's to wrinkled and green, and of course got F1 that showed the 2 dominant traits. Okay, and so we're just going to look at these briefly. To think about the gametes. So now he took these individuals and allowed them to self-fertilize. And so what I would like you to do is, there will be 4 different combinations of gametes possible from this. To just take a look. This is what the chromosomes would look like in meiosis 1 with pairing between the homologs. And just predict the combinations of gametes from these, okay? And remember that there's no preference for any 2 particularly to go together, okay? Okay. Now, these individuals that are heterozygous for 2 different genes, this is the way we do the notation. This is the way we write it. Genetic notation is like a language, so that I could read a cross that was written by somebody in another language, but I would still know from the notation you know, there are two genes and they're not linked to each other. They're on two separate chromosomes, for example. And the term for individuals that are heterozygous for two genes is dihybrid. So di means two, and hybrid is the word that was used in the past, and we now call them heterozygous. Yeah, and so the idea is if there's a gamete formed and it gets the dominant A allele, 50% of the time it should get the dominant B allele with it, but the other 50% of the time it should be combined with the recessive B allele, and there should be no preference for any one of these combinations. These are physically separated from each other. They're on two completely different chromosomes, and so there's no reason why you should have, you know, this is the commonest misconception, is that somehow those dominant alleles are always going to go together and the recessives are always going to go together, and that is not correct. The dominant can segregate with the recessive for the other gene and vice versa. So all the combinations that are possible occur with equal likelihood. And I'm going to make a, and I know for some of you, you're thinking, oh my God, this is so obvious, like why do you keep going on? It's because in the past it has always been a source of confusion, because I think students are not that used to thinking in this way, this sort of kind of thinking. So I'm going through it kind of slow, but just bear with me. So this is Mendel's first law, equal segregation. You've got an individual that's heterozygous. Equal segregation is kind of not really relevant if an individual's homozygous, because all the gametes are going to get the same allele. It's only when individuals are heterozygous that we're concerned about it. And during meiosis, half of the gametes on average should get one of the alleles, and half of the gametes should get the other allele. And I think that the analogy to a coin toss is clear here, right? You have, this is my sad drawing, but half the time the coin will come up heads, half tails, and there's no preference for one or the other if it's a reasonable coin and the person knows how to flip it correctly. And the same with the gametes. So I think there's a clear analogy here. But if we're thinking about independent assortment, now we're actually flipping two coins. Okay, and so for example, suppose one of the coins, we could make an analogy to being heterozygous for the yellow peas versus the green peas, and for the other one we could say, okay, let's just imagine this is the purple flowers versus the white flowers. And so there's a 50% chance, a half chance that the first coin is going to be heads, and a half chance that it's going to be tails, right? And then the second one, there's a half chance of being heads and a half chance of being tails. Half chance of being heads and a half chance of being tails. So if a gamete gets the head, the first coin comes up head, and the second coin comes up head, there's a half times a half, a 1/4 chance of that combination, and then a 1/4 chance, I hope you can see this, for all of these combinations. There are 4 different ways the coins could land, and they are going to be equally likely, right? And so just think about how that would translate to the different alleles, the dominant versus the recessive allele here, and the dominant versus the recessive allele here. And I invite you to just write out what the genotypes would be here, just as a kind of practice. I put the first one in, just as a, when we're talking about the gametes, we tend to write them like this, with just, I mean, obviously just one allele each. And I usually put a comma or a dot or a space or something in between them. That notation isn't of great concern. Okay, so that shows you the alleles of the gametes and the combinations that we would expect to see them. Okay, and just a reminder, going back to the round versus wrinkled, these are the two genes, and in the first sets of experiments, Mendel determined that the round and the yellow were the dominant traits, and you had these 3-to-1 ratios in the F2 when you allowed the F1 to self-fertilize, and that was the dominant to the recessive trait. Okay, now I want to visualize what happens when we have dihybrids for both two genes, and we allow them to self-fertilize. We look at the combinations. Here I've put semicolon, which is probably the best way of designating the genotypes of the gametes. And these are the frequencies, so you can compare these with the ones that you put in for the yellow versus purple. It will come out the same. And so when we do the Punnett square, of course, there are 4 different combinations of gametes. So there's 4 across and 4 down, so 16 boxes in the square. But then you fill in the Punnett square the same way you did with just a single gene cross, except that you're filling in, obviously, more boxes. And because all of these gametes are equally frequent, all of these gametes are equally frequent, everything inside the box, each of the squares, is an equally likely outcome. Okay, excuse me. And when you do that, you can actually then figure out what the phenotypes are. And this ratio is an important one for you to remember, but then to be able to explain how it comes about. So the ones that are going to have both dominant traits, we can identify. There's a picture, of course, but we can identify it based on the genotype. Should be underlining the genotypes here. Sorry about that. Oops. So everything with at least one dominant R allele and one dominant yellow. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9/16 have the 2 dominants. And then these ones are all round, but they are green. So they have the dominant for one, but the recessive for the other. So that's these ones. And then these ones here, they have the recessive for the first one, but the dominant for the second one. And there are 3 of those, 3/16. And then there's only just one that is recessive for both. So you have probably heard of the 9-to-3 to 3-to-1 ratio. 9/16 is both dominants. 3/16, one of the dominants, but the other is recessive. 3/16, the other one is recessive, but the second one is a dominant. And then 1/16 for all the recessive, both recessives. And it would be good for you to remember the ratio because it will save you time, but you should also be able to explain it or use a Punnett square to calculate it or to demonstrate that it's a correct ratio. Okay. And so this is what we call a phenotypic ratio. Now there's more genotypes in there because the yellow is listed this way. So that means some of them are homozygous for the dominant trait, some of them are heterozygous. So this is not a genotypic ratio, but it is a phenotypic ratio. Okay. Yeah, and so it's really important for you to know and understand this. And it's important also to note that this is really just the juxtaposition of 2 3-to-1 ratios. Because if you look at yellow to green, we still see a 3-to-1 ratio. If we look at round to wrinkled, we see a 3-to-1 ratio as well. So this is just the 2 different 3-to-1 ratios, but because the assortment is independent, you can combine them to get the ratio that we see here, 9-to-3 to 3-to-1. Okay, and I've just done this, I've color-coded, which I hope will be helpful. It's easier than a Punnett square to just do some multiplication, and if you are given a cross that is self-fertilized, which is that, self-fertilized, let me do the notation correctly, then you basically just take whatever the ratio would be So Y. For what this cross would be, and it's going to be 3/4 round and 1/4 wrinkled, and that's shown in this column here, right? And then the second thing you look at is this cross, as if it was being done independently, separately, which it sort of is, and that's shown here. So the yellow and the green, and that's shown here. And then we just multiply those individual probabilities. So the chance of being heterozygous, or not heterozygous, having at least one dominant is 3/4. At least one dominant is 3/4, so it's just 3/4 times 3/4 to give us 9/16. I should have color-coded all these. I'll go back and try to color-code them fully in your notes that I post for you. I might have already posted them, so I'll fix them. Chance of having at least one dominant R is 3/4, but only a 1/4 chance of being homozygous recessive, so we multiply those two together, and so on. You can go through and prove to yourself that the 3-to-1 and 3-to-1 ratios, when combined, are going to give you this. And when you do the crosses, this is exactly what you get. Mendel got these kinds of ratios, and so it was a demonstration that his idea of particulate inheritance was correct, and that the different genes segregated without any reference to each other. They did so completely independently. Okay, and I'm going to stop here. There's more to discuss, but this is a good chance to take a quick break, kind of go through the stuff in your head, and then get ready for the next one.